part?
Your little,
That part.
you're fine like that.
Okay. And we gotta do induction and refresh. So I could go through all your PowerPoint slides, but we don't have much time, and I think I'm faster without the PowerPoints. So you can look through the PowerPoints on your own. Some people understand PowerPoints better, some people understand notes on the board better. I don't know. All right. Let's look at our cell. And that's our DNA, okay? I'm gonna draw just the DNA part, if I can. Make a nice flower power. Okay. This is a simplified nucleoid. You know, the nucleoid is a big wadded up hole like this. It's much bigger than this. But I have to draw it small so you can see what I'm doing. I can't work in that whole big thing. And don't forget, you got all your little hydrogen bonds holding the loops together. Oh gosh, I'm getting sloppy with my dotted lines. Whatever. Okay, here's my cell's DNA. Remember, it's the genome. If there's a plasmid, plasmid counts too as part of the genome. But we're not going to focus on the plasmid, we're just going to focus on the nucleoid, okay? Now, let's get back to her question, whoever that was.
What's about, if you have...
Starts and stops. Let me get a couple colors here. Okay, starts and stops. So, how many genes are in a bacterial cell, first of all? Many. Not that many. Compared to humans, not that many.
Wow.
A plasmid can have, say, 5, 10 genes. Bacteria can have some, maybe as few as 100 genes, but probably up to like 3,000. That sound right, Colton? I'm never good with numbers. If I was good with numbers, I'd probably be rich today. But I can't remember numbers. I don't have a mind for numbers. Faces I can remember, but not numbers. And how many genes do we have? Oh, they tell you in base pairs. We've got like billions. But at any rate, I'm only going to draw a few genes. My point is I can't put 1,000 genes on here, okay? But every gene has a promoter and a terminator. And the promoter and terminator is actually a sequence of letters in the DNA, which is a chemical molecule. So what I do for a promoter and a terminator, instead of putting the letters, I just put a block like that. So that would be the promoter, say for gene 1. And gene 1 has a functional part that says put leucine first, alanine second, valine third, threonine fourth, whatever. This is the functional part of the gene. So it might be the functional gene for, say, lactase, an enzyme, something like that. And then it's going to have another sequence that signals the end. That's the terminator for gene 1. Okay? I'm not going to write it for every single one. Just know that then there's going to be a promoter for gene 2 and a terminator for gene 2. Then we'll put a third one, promoter for gene 3, terminator for gene 3. And then don't forget you have your functional part and your functional part. And we'll do one more, gene 4. Okay. Why did I bother to do all of this? Because in transcription, the cell is going to make an RNA copy of a gene, one gene at a time. Not really. You probably need multiple proteins at overlapping times the cell's DNA is gonna be busy. We're gonna photocopy this recipe and make that protein, and we're gonna photocopy this recipe and we're gonna make that protein, and we're gonna photocopy this. But in order to explain it, it's easier to look at it one gene at a time. Yes, Anita?
Is it true that in bacteria, unlike in prokaryotes, eukaryotes, excuse me, you can have translation, transcription at the same time?
Probably more complicated cells, yeah.
Because we
Different things are happening at different times, because the cell has to function. And depending on what signals are there, it might make it start transcription for this one while it's still doing translation for that one. And sometimes certain proteins have to interact with each other, so you have to make this one and then that one. Does that help? So, what's the matter?
Yes.
Is it good?
The thing is, what I was learning in biology, okay, in my case, is that if we take, for instance, a human, how they make proteins, is that the transcription and translation does not happen at the same time because everything is in nucleus. The transcription happens in the nucleus. This thing leaves and then we're going to do translation. In bacteria, in this case, it can be at the same time. You pretty much can have a transcription and translation, and you have another gene 2, for instance, doing transcription
Yes.
and have a gene 1.
In bacteria, they can transcribe multiple genes at the same time and link them together. Yes. It's a little more complicated to explain that, yeah. But for humans, it's a more separate event. Each protein is separate. But in bacteria, they can make multiple copies of the same protein, and they can do different proteins at the same time.
But why is that? Is it because it's one cell?
Because God made it that way. We're lucky to know that it is like that. Never mind why it's like that. I don't know, because actually, bacteria, although they're simple compared to eukaryotic cells, they're more highly evolved. If you think about it, the smarter way is to be simple. So they've had more and more generations than we've ever had. They've been on Earth longer than anybody, and their generations are shorter than anybody. So they've had a lot of opportunity to evolve into the very most efficient way of living. So maybe that's why. Because they're just smarter than us, they're more evolved than us. They've got mechanisms that work that way. I don't know. That's when you know, a good scientist sets up his experiment and then he gets down on his knees and prays to God that it comes out the way he wants it to. Because, you know, scientists aren't supposed to believe in God. But scientists can't explain everything, so that's why I believe there is a God. Because I can explain every nerve and muscle and electrolyte and fluid in her whole body, and I still can't tell you what makes her alive. Because she can have all of that and be dead. So what is the life force, really? Not just air. There's something else that makes all of that stuff work.
A soul?
Your soul, yeah. What is that?
And that's why we have religion, because we still can't explain what the life force is, or where it comes from. Colton's looking at me skeptically. What? All right. Did you ever watch your pet get euthanized?
Oh.
Or watch somebody die? It's like 1 minute you see in their eyes they're alive. And within a split second you see the life leave their eyes, and it's just a blank. You actually see the life go out of the eyes. That's the thing, that's the instant that you can't explain. You know, what is that spark, that life spark? I don't know.
The body doesn't wave us as well.
Who?
Your body waves us after you die.
Oh, I didn't hear that. I didn't know that.
So what our soul is in us but it's invisible.
going to talk about operon theory. And this is still the genetics chapter, okay? So this is genetics chapter 7. Okay. With DNS. All right. Operon theory. Every gene is called an operon. So let's look at this green one right here. Okay? The operon, and we're going to simplify. Your textbook gets into some hairy details that we don't have time for or really care about at this point. So, an operon is a gene, the functional part of the gene, plus its promoter and terminator. If we look at bacterial cells, their genomes are much more simple than eukaryotic genomes. So it's easier to study it in bacteria. But why do we even care about all of this? They're attempting to explain cell differentiation. Okay. Here's my dad and my mom. And one fateful night they get together. She drops an egg, he drops a sperm, fuse together.